Good morning, I am Ayushi from group 319. My today's topic is conformation of the polypeptide chain. To understand the protein structure, it is necessary to consider the possible conformation of the polypeptide chain. These conformations are primarily determined by the structure of the peptide bond. The peptide bond is CONH. The structure of the peptide unit established by means of the X-ray analysis of the peptide and related compounds are given in the table 4.1, and the table 4.1 is given below. This one is table 4.1. The full extended chain without deformation of the valence angles and change of the bond length has a trans conformation with zero values of the rotational angles A, B and C. Such a conformation is not, however, the most stable. The hydrogen atom of the amine group NH form hydrogen bonds with oxygen atom of the carbonyl groups. Like here, the carbon atoms are forming the bond with the hydrogen group and also with the nitrile group. In order to find the most stable conformation of the polypeptide chain, it is necessary to minimize its total energy, including the energy of the intramolecular hydrogen bonds. Pauling and Corey have found the most stable conformation of the polypeptide chain on the basis of the data obtained by X-ray diffraction studies and of the consideration of the tight packing of the chain with the maximum number of the hydrogen bond. There are three such conformations. First one is alpha helix. It is characterized by what the rotation about the axis by 100-degree angle and the displacement along the axis by 0.15 nanometer per peptide unit. Accordingly, there are 3.6 peptide units per each complete turn of the helix, as seen here. The height of the each turn being 0.54 nanometer. The helix is stabilized by the hydrogen bond formation between the carbonyl and the oxygen group of the one peptide bond and the hydrogen and the nitrogen group of the preceding fourth peptide, and such bonds are formed between all the amino acid residues with the exception of the proline, which contains no NH group. That is, it doesn't contain any bond between the NH group. It only contains the bond between the carbonyl and the oxygen group and with the hydrogen atoms, but not with the nitrogen and the hydrogen atoms. The alpha helix may be either right or left-handed, but mostly it is the left-handed structure we are known to. The second and the third conformation with maximum saturation of the hydrogen bond, parallel and anti-parallel beta form. Here in this structure, this is the alpha helix. Nitrogen makes a bond with the oxygen and with the carbonyl group. Carbonyl is attached to the methyl or the ethyl group with the oxygen group with the carbonyl, then the nitrile, then the oxygen and nitrogen form double bond with the oxygen. Then it forms an imaginary bond with the oxygen that is associated with this carbon atom of the methyl group. These conformations are the conformation not of a single chain but of the assembly of the chain that form a beta plated sheet structure. The chain in the beta form have no planar trans structure. In the parallel beta forms, A is equal to 1 and beta is equal to 22.9 degree. A 11 degree and B is equal to 22.9 degree. And the anti-parallel beta form, A is 380 degree and B is the 325 degree. This is the parallel beta form. And this is the anti-parallel beta form. This is the parallel one in the same direction and the anti-parallel in the opposite directions. The hydrogen bonds stabilize the fixed conformation of the polypeptide protein chain in solution. The fixed conformation is commonly referred to as the secondary structure of the chains. The presence of this secondary structure, which is periodic, means that the chain is similar to a crystal. This is a crystallized structure, a crystal-like form. The alpha helix is similar to one-dimensional and the beta form to a two-dimensional crystal. The alpha and the beta forms are not only ones. For example, fibrous protein have also other conformation. Alpha and beta are not only the ones that are present, but the fibrous protein also, fibrous protein also has alpha, beta, gamma, and so many plated structure. These are only available for the parallel and the anti-parallel form. This is the schematic presentation of the cross beta form. The conformational energy of the chain is determined by the weak interaction of the non-bonded atoms because of the planar structure of the peptide group of the rotational angles xi and the xi1 of the 11th unit are the practically independent of the angles xi plus 1 and xi plus 1 of the adjacent unit. If these both angles vary within the limits, they are allowed with the overlap of the atoms of the peptide group linked by the bonds unit, and if other angles vary simultaneously, then there is no such a combination of these four angles at which the steric interaction of this unit with I plus 2 and the unit is possible. Therefore, the polypeptide chain has limited cooperatively the short range interaction being stimulated to nearest neighbor interaction. This allows one to deal separately with. Then the conformational energies for individual amino acid residues. The steric mass for a particular residue depends substantially on the nature of the radical R. It may be presumed that the interaction is given in the pair of the peptide groups characterize the amino acid residues that links these groups together. Ramachandra, a scientist, performed the studies that the dipeptide glycine 1 alanine and obtained a conformational steric map for the alanine. The calculation was based on the assumption that atoms are rigid spheres having van der Waals radii. They have the van der Waals radii determined from the data on the intermolecular distance in the molecular crystals. Using potential of the rigid sphere, one can evidently obtain only the allowed and the forbidden regions of the values of the given. In figure 4.7 it is showed. It shows a map for the alanine. The solid lines delineate the regions allowed at the ordinary internuclear distance, and the dotted lines those allowed at the minimum distance. Dotted line, these are allowing at the minimum distance, and these are the the solid lines that delineate the ordinary structure. The map gives the value of the xi and xi for the right and the left-handed alpha helix. The map gives the values of these two figures for the right and the left-handed alpha helix, alpha 1 and the alpha 2, the parallel and the anti-parallel beta form, beta 1 and the beta 2, and for the follows medium collagen. Analogs maps have also been plotted for other residue. Naturally, as the size of the radical R increases, the region of the allowed values of the xi and phi diminishes. This is the graph that was explained before. More regions calculation have been carried out with the acid of the following potentials. This is the formula given. The potential U may be taken in the form of the Lambert-Jones potential at the 6 experimental potential. The quantity characterize the column electrostatic interaction between the neighboring pair of the peptide groups, interaction between the neighboring pair of the peptide groups, which is determined by their large dipole moments amounting up to 3.7 diopter. The energy barriers are bound from the data obtained in the investigation of the small model molecule. Evidently, this is the formula or the quantity is given. These barriers are small, much lower than these of the hydrocarbon. The parameter for the Lennard-Jones potential are listed on the table 3.2. The calculation is complicated by the fact that the value of the dielectric constant that appear in the column below is unknown, and the formula given is this one, where E1 and EF are the charges on the atom I and J. If there are no other atoms or the molecules of the solvent between the interacting atoms, then I is equal to 3. The value corresponds to the hydrocarbon of the organic environment. For atoms, carbon and nitrogen linked by a hydrogen bond, use can be made of the function USC is equal to UIJ plus UC, which is determined and form the value of the hydrogen bond energy and equilibrium. And my next topic is the goal of the protein physics. Protein are the invariable participants in all life processes. Protein enzymes catalyze all the chemical, electrochemical and mechanochemical processes that take place in the cells and in the organisms. The most important function that of the protein is enzymes. It catalyzes too much enzymes. Specialized enzymes serves as a catalyst for all metabolic reactions, and these catalysts they enhance the rate of the reaction, or they can also decrease the rate of the reaction. DNA replication, the transcription of the genetic message from a DNA strand to the mRNA, and the transfer of the information encoded in the mRNA to the ultimate structure of the protein molecule. The process referred to as the translation. This process is referred to as the translation. Protein also function as the regulator of the genetic function of the nucleic acids. Regulatory enzymes called the allosteric enzyme. Allosteric enzymes take place in the role of the cascade processes enzymes that takes place in too many processes, provide feedback in the metabolic chain. The movement of the cell of the organism, the performance of the mechanical work by them is realized by the special contractile protein which serve as working substance for these processes. Contractile protein perform the enzymatic ATPase function, accomplish the conversion of the chemical energy stored in the form of the ATP to the mechanical work. The hanging up of the storage battery, that is the oxidative phosphorylation, occur in the mitochondrial membrane with the requisite invariable participation of the respiratory chain enzyme. These enzymes are also catalyzed by the proteins. Oxidation reduction enzymatic processes also occur in the photosynthesis. Other membrane proteins are responsible for the active transport of the molecule and ion through the membrane. It can be through the ATP pump or the oxygen hydrogen pump. And consequently, for the generation and the transmission of the nervous impulse, protein control all the metabolic and the bioenergetic processes. In animal organism, there are special protein which perform specific function. The proteins serve to store oxygen, that is myoglobin, and transported hemoglobin. Low molecular weight protein, or more exactly polypeptides, they are hormones. The gamma globulins of the higher organism protect them against foreign biopolymers by functioning as antibodies in the immune processes. Finally, protein that enters into the composition of the connective tissue, it cartilages and tendons, and also the protein of the skin, hair, and the feathers are the structural protein which provide a reliable and the mobile active intercommunication between the organs and the unity of the organisms, and its protection against the external influences. One can say that protein are invariably involved in the storage, transfer, transformation, recording, and the reception of the chemical signals, macromolecule, molecule, and ion in the living system. In many cases, the protein themselves serve as the signals. They are receptor and the transformers. Protein that enter into the composition of the receptor system of the organism records external signals into the chemical and the electrochemical language. Thank you.